Good morning, good morning, good afternoon, and good evening to everyone joining us from different corners of the world. No matter where you're joining from, whether you're starting your day, enjoying your afternoon coffee, or winding down in the evening, we're delighted to have you with us. On behalf of the Women Fishery Graduate Network, I extend a warm welcome to our distinguished speakers, respected guests, and enthusiastic participants. Before we dive into today's a fascinating discussion, the DNA of tomorrow, let us ask a simple question. How do you think genetics and molecular technologies are transferring fisheries and aquaculture?
Well, to answer this question, we need first to picture the future of fisheries. When we think about the future of fisheries, we usually think of huge ships, oceans, and bulk harvesting of fish. But some of the most important work is happening at a sea level we cannot see, in genes, in cells, in the molecular code that decides which fish survive, which thrive, and which we might otherwise lose forever. DNA based tools improve fish breeding, disease resistance, biodiversity conservation, and sustainable large scale fish production. As global demand for aquatic food grows, these innovations are becoming essential for the future of aquaculture. This is how genetics transform fisheries and aquaculture. With this, let's explore.
Good morning and warm welcome to session 8. My name is Kirstana Balamurgan. Most people call me Ki, and I'll be one of your hosts today. I am a plant molecular geneticist by training with a master's in plant genetics and crop improvement. From the John Innes Center and University of East Anglia in Norwich, I am the Overseas Research Coordinator with the Women Fisheries Graduate Network. My research focused on how plants sense and survive stress at a molecular level. I've always been fascinated by how the code written in DNA shaped the living world, from crops in a field to fish in a sea, which makes this session especially close to my heart.
Thank you, Kirtana. I will be your co host today. My name is Niharani, and it is a privilege to be part of the WFGN Global Interactive Summit 2026. I recently completed my Masters of Fishery Science in Fish Biotechnology, where my research focused on molecular biology and gene expression analysis in fish. My academic journey has inspired a deep interest in fish genetics and biotechnology, making today's session especially exciting for me. I am delighted to introduce today's distinguished speaker for session 8, The DNA of Tomorrow. It is a pleasure and an honor to be your co host today, alongside my wonderful team, Kirtana and Akansha. I look forward to learning with all of you and hope you enjoy this insightful session. Thank you.
Hello to all and wish you a beautiful day so far. I, Akanksha Tiwari, am a PhD scholar from the Department of Aquaculture at Dr. Rajendra Prasad Central Agricultural University and acting as the General Secretary of this prestige organization that brought us here together. Molecular work and bioinformatics have always fascinated me. I've been learning things like gene expression and molecular docking a little to enhance my knowledge in this field. So I am really looking forward to this session.
Um, Ladies and gentlemen, close your eyes for a minute and think about the word DNA. What do you what comes in your mind? For decades, this word belonged strictly to laboratory textbooks. It meant a biological blueprint, a microscopic twist of molecules hidden deep inside the cell, silently dictating the traits of a living organism. But as we look to look towards tomorrow, our global food system are facing unprecedented pressure, and DNA has evolved into the most powerful tools for survival. Specifically, it is reshaping the future of aquaculture. We are moving far beyond traditional fish farming. Today's aquaculture relies on genetic blueprints to breed resilient, disease resistant fish, select for faster growth, and protect fragile marine ecosystems. For understanding and mapping the genetic code of aquatic species, we can optimize nutrition, reduce our reliance on wild caught feed, and create a truly sustainable blue economy. The message is clear. The choice we encode into our aquatic systems today will determine how we feed a planet of 10 billion people tomorrow. We are no longer just harvesting from the water, we are actively programming the future of sustainable seafood. Today, let us explore what it truly means to build a genetic DNA designed for the aquaculture of tomorrow. Let's begin today's session. Over to you,
Thank you.
Neha and
Thank you,
Akansha.
Kirtana. It
Thank
is my
you,
distinct
Kirtana.
honor to introduce our first esteemed speaker, Dr. Sabarishwaran Kandaswamy. He is an assistant professor in the Department of Biotechnology at PSGR Krishna Mammal College of Women, Coimbatore. We are especially honored to have him with us today because he has recognized among the world's top 2% scientists for 3 consecutive years, 2023, 2024, and 2025. Having such a distinguished researcher with us is truly an immense pleasure and a privilege. His expertise spans biotechnology, molecular biology, environmental biotechnology, and sustainable scientific innovations. He has made significant contributions through research publications, books, patents, and various academic activities. His research explores how biology can help solve some of our most pressing environmental challenges, from converting, from converting biomass into clean energy to developing biodegradable algae based bioplastics and using microbes to remediate and restore polluted environments. His work beautifully combines molecular science with sustainability, transforming waste into valuable resources. This vision aligns perfectly with the theme of today's session, the DNA of tomorrow. As fisheries and aquaculture continue to grow, challenges such as improving water quality, managing waste, and ensuring sustainable production become increasingly important. Biotechnology and molecular tools offer innovative solutions to address these challenges and shape the future of sustainable aquaculture. Everyone, please join me in extending a very warm welcome to Dr. Sabarishwaran Kandaswamy. Sir, it is our pleasure to have you with us today. The virtual platform is now yours.
Unfortunately, as Sabriswaran is running a little late, I would like to move on to our next speaker, uh, Dr. Pravesh Kumar, who will be introduced by Akansha. Over to you, Akansha.
Thank you so much, Ki. So, it is my esteemed pleasure to introduce our first speaker, that is Dr. Pravesh Kumar, Assistant Professor at the College of Fisheries in Dholi, part of the Dr. Rajendra Prasad Central Agricultural University in Bihar. Dr. Kumar is a fish nutrition scientist, and his work tackles a problem that sounds humble but matters enormously. How do you keep baby fish alive? Take Magur, the Asian catfish, a prized nutritious food fish across much of Asia. Farming, it is held back by staggering mortality in the first days of life. Dr. Kumar has studies how enriching the tiniest live feeds with essential fatty acids and vitamin C can help those larvae survive and grow. A small intervention with a huge consequence. Because more surviving larvae means more seed, more farms, and more food. More recently, his research has moved into a fascinating idea called early nutritional programming. The discovery that what a fish eats in its earliest days can shape its health and huge performance for the rest of its life. He has pursued this in collaboration with researchers in Hungary, and he is exploring how to feed fish more sustainably, testing alternatives to expensive fish meal from plant byproduct to nanoscale minerals. This is science with mud in its boot, the kind that quietly underpins food security and the livelihoods of thousands of farmers. Please join me in welcoming Dr. Pravesh Kumar. Sir, the virtual platform is now yours.
Thank you, Akansha. Am I audible?
Yes, sir, you are audible.
Okay. Thank you so much, first of all, for inviting me on this platform. I just share my slides. Slides visible?
Yes, sir, they are visible.
Okay. Thank you. Thank you so much for having me. So I joined one of yesterday's session, and I hope that most of are here the researcher, PG, PhD student, uh, apart from the scientists across the globe. So I just try to give the gist of my work because the time, what I know, it's very limited. It's only 10 minutes. So I prepare in a way that I can give you the overview. So today, the topic which I choose for my talk is the fish reproduction and aquaculture, translating the molecular research into the practical applications. So if we see in the reproduction or in the fish breeding. So the HPG axis is the major axis, which give the information or the clue for the breeding, have the different sort of the genes, neuropeptides, hormones. And in those genes and the hormones, GnRH is considered as a one and most prominent hormone, which is related to the reproduction. But if we go back for 1 or 2 decade backs, there are the certain other, especially the neuropeptides, which also show their potential in the uh, regulation of the reproduction, especially in the fishes, like kisspeptin, GNIH, ghrelin, leptin, and oxytocin. So these are the different type of the neuropeptides, which also have the different impact on the reproduction. So today I will focus on the work which we have done here. That the GNIH. GNIH is a gonadotropin inhibitory hormone. So the name itself says that it is a inhibitory in nature. And we have, when we go for the literature, and we have observed that it have the different role in the different type of the groups. Like in fishes, it have the both inhibitory and the stimulatory effect on the reproductive axis. But in the birds and mammals, it show the mostly the inhibitory effect. So in the nomenclature also, it's not the same throughout that they are called the GNIH. In amphibians, it is called the different in mammalians with the different names. And if you saw in the right side in the table, that it have the inhibitory effect in some of the fishes and stimulatory effect on the some of the fishes on the reproduction. But there are a species where it show the both type of the impact, like in a single species of goldfish, it have the stimulatory and inhibitory effect also. So when seeing that, we also think that, okay, we have to check for the IMCs, like our Indian major carps. So we choose the Katla for the research, and we saw that it is GNIH have a specific functional diversity, and we characterize the GNIH system, we study the expression, we study the docking, and in the last we do the functional validation. So here is just a pictorial view of the model of the GNIH receptor, which we develop, and then we check the docking analysis, to know that how it bind to the GNIH, so that it is having the proper binding or not. And then we check the expression of these genes throughout the different tissues which have the potential type of the tissue where this type of the GNIH can express. And we find out very interesting thing that these genes or GNIH or their receptor are mostly in the brain or in the gonads. But if you see the last one, that is the GNRH receptor 3, which is exclusively present in the gonads, not in the brain. We saw that this hormone, this receptor, is also have the very much role in the gonads also itself, like in the process of the steroidogenesis, not like from the brain itself. So it is also the primary sites for the axon. And we observed that during the annual reproductive cycle, from preparatory phase to pre-spawning, spawning, and spent phases, in these species, exclusively in the cutla, we have observed that it is mostly observed the higher level was detected either in the spawning or in the spent phases. So it so that it can have the dual effect in the cutla itself. And when we have observed that, okay, so we have to check how this can have the role. So for that, we choose a antagonist which have the antagonistic effect on the GNA, and we also take the like a same type of the hormone which have the stimulatory type of the roles. So this antagonist so that it can have the stimulatory type of the effect. If you can see that at the initially the level of the hormones like the FSH, LH were the higher, and when we give this antagonist, it will reduce. And for the GNIH, it show the higher expression of these hormones. So like same here that we used the GNIH along with the LHRH. So to know the combinatorial effect. And we observed that, okay, if we give in the combination, that it enhance the expression of the different type of the genes and also the hormone level of the reproduction related hormones. So we saw that during that particular pre-spawning phase, which we have seen in the Katla, it have the stimulatory effect. And we also check some of the like the negative regulators of the production, that is the melatonin. And if it is combined with the GNIA, then the expression level of those type of the hormones will be reduced. So in the last, we want to say that in the Katla, this GNIH have the stimulatory effect during the preparatory or pre-spawning phase. So we have to check further in the other species or of the IMC, and also during the different seasons in the Katla also to confine or to say that, okay, this is the role. And these are the few papers which we published from this work. Now we move to the one another hormone or the gene which is called the star gene. Star is a stereogenic acute regulatory protein, which is a precursor gene of the cholesterol synthesis pathway, or specifically we can see the steroidogenesis pathway. Because in the steroidogenesis pathway, it is much more related to the reproduction, where estradiol, progesterone, or whatever the hormones which are helping in the reproduction are produced through that pathway. And for that, we have a develop a construct. For that, we have developed a construct called the expression construct of the star gene in the PCD and a fourth expression vector. And we transfected that into the SS1 cell lines. And through the Western blotting, we have shown that the star gene expression is started, and it's showing the stereogenic genes expression is upregulated. And this study we have done in the Claris Magur, where it showed that the genes of the different reproduction genes are the enhanced if we give the expression construct of the star gene. So by having all these molecular related research, we move to the breeding of the Magur, and in that one we have to that the how this fish can be breed, and we have checked the breeding parameters. We did this flow type of the hatchery, we have makeup, and from that, and in the last also we have gives a vitamin enriched live food infusoria, and for that we obtain a paper also. And now currently we have conducted some work on the breeding of the Mestra circulators, which is commonly called Gechi here, and we conducted the breeding with the help of the different hormone doses and the different type of the hormones. and we successfully breed it here by the different stages, and from that we publish a paper and release some technology for this induced breeding of the Getchi. And if we go to move forward, so we here is a one slide which I try to give the glimpse that now we are working on the stress, transportation stress, animal welfare also, so that this different type of the essential oil which are present from the different plants to check their bioactive profile and to check their aesthetic effect and to know about the animal welfare aspect of that different type of the oil. So we are assessing it and we are trying to find out in terms of the animal welfare, and also we will try to do in the breeding also that how they are. A few experiment we have conducted this and few are under the process. So in the last, if I conclude that the research what we are trying to hear that from the molecular endocrine or reproductive biology to the sustainable aquaculture for the breeding of the fishes, for the sustainable production of the fishes, and for the welfare point of view also. So that's all. Thank you so much. If any doubt, any things are there, then you can contact. Thank you so much. I hope I finish on the time.
Thank you, sir. Your presentation has shown us that the future of aquaculture is not shaped only by advanced genetics, but also by thoughtful nutrition and innovative hatchery science. Your research reminds us that even the smallest interventions in the earliest stage of life can have profound impacts on sustainable fish production. It has been a privilege to hear your insights today. On behalf of WFGN, we thank you for an outstanding presentation and for inspiring us all.
Thank you. Thank you so much.
Our final speaker takes us to the very close of today's theme, the genetic code of fish themselves. I welcome Dr. Suresh Eswaran, sir. He is the assistant professor and head of the Department of Fish Genetics and Breeding at the CIFE at the Institute of Fisheries Postgraduate Studies, Tamil Nadu, Dr. J. Jayalalita Fisheries University. He completed his PhD in fish genetics from ICR CIFE, Mumbai. His research focuses on fish genetics, DNA barcoding, molecular markers, and genetic improvement for sustainable aquaculture. In practice, this means using DNA barcoding to identify species with precision, molecular markers to guide smarter and faster breeding, and population genetics to reveal the hidden diversity within our fish stocks. These are the very tools that allow us to conserve biodiversity, combat seafood fraud, and breed healthier, more resilient fish for the farms of the future. It is difficult to imagine a more fitting expert to close the DNA of tomorrow. Everyone, please join me in welcoming Dr. Suresh Eswaran, sir. Sir, the virtual platform is now yours.
Yeah, thank you, Neha. Thank you very much for a nice introduction. Of course, I have done little work only. Dr. My friend, Dr. Pravesh Kumar, he has just presented. Yes, I have been working on fish genetics for the past 15 years. Specifically, my specialization is population genetics or population genomics. So I started my work on the fish called mullet, that is a practice water fish, Mugilcephalus, and followed by macrobrain rosenbergi, then some of the shellfishes, that is Pinnaeus indicus. Specifically, it's not single fish, just I look the fish species as a whole population, like how human beings are there, different populations are there. What kind of genetic variations are occurring in the population level using DNA molecular? Basically, I can say I work on molecular marker, that is specifically DNA marker. There are many markers available. You must have heard, like it started with the protein marker, biochemical marker, followed by nuclear marker, that is RAPD, RFLP, AFLP, microsatellite, currently SNP, all these markers. But I have worked all these markers. Currently, I'm working on SNP markers also. So this is very important because see, this any population is evolving means it should adapt to the cope up with the environment. Otherwise, that population will not survive. Specifically, if you heard this Hardy-Weinberg equilibrium, the population should evolve. If the population is not evolving, means, as for example, the human being now, it should look like ape only. Therefore, population is evolving. Then if population is evolving means what is happening? What are the factors that are responsible for the population evolving? There are many factors that specifically it is a mutation, migration, and selection. Whether it is happening or not, if you ask me, it is happening. Without these parameters, any population will not evolve. If we believe that human also evolve, the fish population also are evolving. That is the reason the different populations adapt to the different environment. For example, the reason the pandemic that COVID came, most of the people were affected, but some of the people were died. Some people affected but survived. Some people did not affect. Why it happened? It is about the DNA. The DNA that is located on that particular population. Those who adapt to this changing environment are surviving now. For this adaptation, the underlying DNA is very important. Whether those genes have resistant or not, that resistant gene can be founded only if there is a variation. There is population level variation. If there is no variation, there is no evolution. If there is no variation, we cannot improve any fish in any selective breeding program. The basic parameter is genetic variation. If there is a genetic variation in the population, then only we can go for selective breeding program, marker assisted selection breeding program. So far, I have used microsatellite marker. Currently, we are using this microsatellite marker for estimating genetic variation in the Scatophagus argus. That is one of the fish called brackish water fish that is used as ornamental as well as food fish. That is sponsored by DPT. Just now we started. Then move on to the next point, that is it's a DNA barcoding. DNA barcoding, specifically, we use this technique for identification of any fish species, but especially you must have heard the seafood is one of the very important economic activity worldwide. Out of all these, at least 30% of the seafood or it is estimated that 20 to 30% seafood are fraudulent. That is either substitute or different species are. For example, if you want to eat fish, if you go to any restaurant, if you order for share fish, the cost is around 1,000 to 2,000, the various prices. But instead of that, if you are given the fish tilapia, you don't know which fish is good to you. But you are finally, it is huge loss for economic activity for the consumer as well as for the country. That can be detected. If you what fish you are served in your plate, you should know. That can be detected through DNA barcoding. That has been done around over the world, but few studies have been done in India by Nagalishmi et al. Also from CAPC has done. She has found that more than 30% of the fishes are fraudulent, substituted. Even we have been working, our Tamil Nadu government has sponsored around 25 lakhs for that project the last year. This year, March month, we have to work on that. What is the substitution rate in Tamil Nadu? That we collect the fish sample. We will use this molecular marking to identify which fish species are substituted, what is the rate of substitution, that we keep on working. Then another one is we will go for this one meta barcoding also. Meta barcoding, for example, in the feeding ecology, if you want to maintain any food chain value, we should know the feeding ecology. For feeding ecology, for example, so if you use the traditional technique, what fishes are eat, if it is a carnivorous or herbivorous water fish. If you analyze the gut content, you have to see under the microscope and what is available in the food system. You have to check and you have to see. That is very difficult nowadays. But using the DNA marker, that is meta barcoding, you can analyze what are the species available in the gut content. So therefore, the value chain, or this means food chain, can be analyzed. The ecosystem can be managed through DNA meta barcoding. So these are the areas we have been working. And in addition to that, aquaculture point of view, we have taken this one of the classical technique or classical breeding program, that is diallele crossing program. We have used 3 different strain tilapia, that is gift tilapia, as well as tilapia from red tilapia. Another one is imported tilapia. These three tilapia we have crossed in the experimental design. 3 into 3, 9 experimental design crosses. Finally, with the triplicate, we have got 27 breeding pairs. Out of this, we are trying to improve one new breed or new local tilapia that can be sold for the aquaculture point of view. Even though all over the world tilapia farming is famous, even we should have a tilapia strain that is adapted to the local environment. That is what our aim is. So we have last year we have completed. We have founded one strain, but the color is it is between night tilapia as well as red. Therefore, that is what we are working on it. That is giving very good results. So these are the area we have been working, but the next level we would like to go for the marker assisted selection, because the classical selective breeding program takes at least 10 to 15 years to develop one species for genetic improvement program. But through molecular assisted selection, marker assisted selection, you can reduce the time period within 3 hours. Within 3 years, or any program we can finish it up. Because in that also we use any DNA marker only. So the base for all these workers is it's a DNA marker. It can be either mitochondrial marker or microsatellite or a SNP marker. These are the one we have been working, but we would like to go for a long way. And one more important problem currently in the tilapia is it's a mixed sex culture. Different sexual growth is there. For male grows faster than female, therefore farmers prefer all male population, all male they need. For that, the current practice is using the hormone. So the hormone practice is not advisable. Many people, many customers don't prefer hormonal admitted population. Therefore, we are trying to use the DNA markers to our gene editing tool to identify that the gene which is responsible for all male. We would like to develop 100% all male tilapia without hormone, without using hormone. That is our next aim that we are currently working on it. These are some of the area on fish genetics, and we have been working. Thank you very much for the opportunities. If you have any questions, we can clarify in the panel discussion. Thank you so much.
Thank you so much, sir, for your insightful talk. Your insights into DNA barcoding and molecular markers have shown us how molecular technologies are revolutionizing fisheries and aquaculture through accurate species identification, genetic improvement, and biodiversity conservation. Your expertise has given us a deeper appreciation of the role of genetics in shaping the future of sustainable aquaculture. We sincerely thank you for sharing your valuable knowledge and experience with us today. It was truly a pleasure and an honor to have you with us. Thank you, sir.
Thank you. Thank you so much.
Thank you to all our speakers. Those were wonderful insights. I would now like to open the floor to our audience. If you have a question, please use the chat or raise hand feature and I'll bring you in. Do let us know if your question is for a particular speaker or for the whole panel. While the first question comes in a gentle reminder to keep questions brief so we can include as many voices as possible. Let's begin.
Dr. Suresh Eswaran,
Dr. I think Sagayas Victor has question for Dr. Suresh. What do you want to ask, sir?
Yes, please.
yeah,
Go ahead.
really thankful because you narrated the way you narrated for metagenomics, how it is useful. I mean, genetics useful to find out the gut feeding behavior and everything, and why genetics is important in 10 minutes. Really is amazing.
Thank you so much, sir. Thank you so much. Otherwise, if I knew that, I would have prepared much more to share my knowledge. It was just whatever I could come to my mind, just I shared with you. So thank you so much.
Because earlier we don't care about the gut, but you narrated that why gut genomics are very much important, because we know the population and we know the microbiotas, and then the gut is an important thing for aquaculture. If gut is failed, then everything is failed. So the way you narrated about the wild and the gut health and everything, and apart from this, I personally noticed that about here in Tamil Nadu there is shearfish, Vanjaram.
Yes.
So used to mix with koala and vanjaram. They sell cola
Yes.
and vanjaram. Cola is a lower value, but it belongs to the same family. It's both on the
Yes.
sea fish family. So it's
Yes.
right? Combromidis. But
Yes.
they separated. The original CFP is 1,200 rupees today in the market, but the cola we call Aracola, right? Aracola is around 600 rupees, half of the price. But
Yes.
if we go to the shop, they sell as
Yes, sir. Mostly
yes.
they
Thank
sell
you,
it
sir.
Barsha, this Pangasius fish. They actually the Pangasius fish it is substitute for most of the high value fish because the filleting is very easy in Barsha fish. They just did so 100 only per kg. They just replace with this fish. Once it is cooked, nobody knows which fish is this. Just you have to go even fisheries who studied fisheries in PhD level also are cheated by these restaurant people. It's a very serious threat to this. So that work also we have taken up, sir. Yes. As you said, such a very that gut analysis is becoming a very important research all over the world. If you fish, you take in aquaculture, you take out with you look at the papers published in aquaculture on the topmost channel. The mostly now it is a gut. Gut is for everything. If your gut is including human being and animal, your gut is good, then everything is good. Gut is connected with the brain, liver function, any function. Therefore, the gut microbiota and gut content analysis becomes a very important task. Therefore, in the future, any genetics and biotechnology or any advanced technique or anything, we should concentrate on our gut level also. Yes, sir.
So one more question, sir. Do we have a SNP
Yes, sir.
panels for our IMC? Do you have
Which one, sir?
SNP single nucleotide polymer?
Yes, sir. SNP
Do we
panel
have
as a
a
panel,
SNP?
they got. Yes, sir. SNP, no? Single nucleotide polymorphism.
SNP, yeah. So do you have it for IMC? Katla road and
Yes, sir.
all?
No, no, we do.
Because
We means they have identified what are the SNP, but as a panel based on the we can develop SNP chips based on that. But currently we don't know. But we can ask the company people to give us if we give the details, they will give us.
because we are not getting a qualified seed, sir. If SNP is
Okay, okay.
created,
Yes, yes, that is quality seeds
we,
is one of the important problem genetically specifically, and that's what that they always suggest that certification, quality certification, seed certification is very important in this aquaculture. So we are also trying to establish one seed quality certification system, specifically without inbred. So how we can determine we can give that is also in one way we are planning. Sir.
okay, sir.
I have a question. It is for the entire panel. As climate change alters water temperature and introduces new diseases, how can we use DNA mapping to breed fish that are naturally resilient to these environmental stress?
Yeah, I can some throw light on that. That means, see, this temperature is changing. There is no doubt. If while changing the temperature, those people, as I said just sometimes back, the adaptation is more important. That is called natural selection. Those who adapt to that environment only will survive. So, what we have to look at that fish and control, we can say those fishes that are not adapted. We need to compare these two entire genome level or transcriptome level. We have to check the one that is adapted to that coping the environment or changing environment should be identified first. That should be developed in the captive environment. Just maybe available in the wild, but it will be just survive in the wild level. But once you develop the brood stock or whatever the seed quality technique in the captive level, that can be only used for aquaculture purpose. Otherwise, it is a natural only. Therefore, it can be identified, but it needs a lot of facilities and the effort and cost. Yes, it is. We have to go for this next generation sequencing. We can identify, as Sir said, we can identify the SNPs also. We will get thousands of SNP. Out of that, we can narrow down which SNP is responsible for the changing environment that is adapting to that temperature changes. We can identify those fishes can be selected for the future generation or for breeding purpose or aquaculture purpose. Yeah, some other can be added.
We have another question. This is from Aditi. She's asking, as metagenomics detects DNA from both pathogenic and non-pathogenic microbes, how do we detect which is the primary pathogen causing a disease?
See, this with metagenomics, yes, it detects whatever the DNA available, it will detect. So it is just we will have already the database, which what are the pathogens and microbes are causing disease. So whether those disease causing organism DNA available in the data or not, we need to check. It is first finding, then we cannot find it. It's a reverse case. We should have already the report that the organism which causes the diseases. Then we can detect from the data. That is how it is. We can do it.
Thank you,
Hello.
sir.
May I ask the question, please?
Yes, you can.
Uh, Sir, I am the one I was asking this question. Sir, it is such that Aeromonas and other bacteria are opportunistic in nature. They are usually present there. And if there is a fungal, just say there is a fungal infection as well, how can we then determine that the primary pathogen mainly is that fungus, not the bacteria, I mean?
That means, if I understand the question correctly, you want to know whether the disease is caused by Aeromonas or fungus, correct?
Yes, sir. Or other viral diseases also, if they are present. I mean, the bacteria such as Aeromonas, they are usually present in the biota. I mean, they are the opportunistic pathogen. So sometimes they are secondary invaders, and the primary are the viruses or some other bacteria, maybe. So, in a set of that metagenomic DNA, how can we then determine that which is the primary one?
No, only the same that as I said just now. No, it is it. This is through experiment only you can do. Just you cannot directly identify it. First, you have to infect the fish separately and together only. It's like experimental design T1, T2, T3, like we have no. Like that, we should have
Yes,
different
sir.
kind of setup. In that aeromonas only, and the secondary one, what you will say, and control without that. So then all the three we have to do for metagenomics. Then we can compare which gene is expressed, which one is expressed. From that we can identify. That is how it is we can do. We can identify. But directly from the infected fish, if you take the data and we cannot say. But we need to do the experiment at the wet lab level.
Okay, so thank you, sir.
Yes.
Also,
Yes.
I had some conceptual confusion between eDNA and metagenomics. So,
Okay.
what is the difference?
So yeah, it is more or less means intermittently it is used. EDA can be anything. It's an environmental DNA. Okay, this mostly this eDNA, the samples are collected from the water soil sources. In that, fish's feces or mucus will be settled. So when fish moves, the mucus settle down, and any slime or anything settle down. From that, we can collect the water samples we are collecting. We are filtering. We will get some DNA. From that, we can isolate the DNA. Then we do experiment. This is environmental DNA. Metagenomics generally talk about once at the gut level, we can take the entire samples from the one particular place. Entire samples we can take. If we go through that, if you do the analysis, that is metagenomics. Generally it is also environmental level we can take, but mostly environmental DNA is you can take anything, including soil. That is how it is.
Yes, thank you, sir.
I have another question for Pravesh sir. Um, your
Yes.
work is mainly on early nutritional programming. No, sir. How should we change the hatchery practices for Indian carps and catfish?
Yeah. Not exactly nutrition programming, but yeah, I am much more working on the reproduction and breeding aspect. So the thing is that your question is very valid, like specifically, not in the IMC, but if we say in the catfishes, the early nutrition or the larval rearing is very, very important aspect, because if you see these are the fishes which are having the uh, carnivorous in nature. They are showing the cannibalism, they are showing the heterogeneous growth. So we have to much take care about these fishes. So for that, this early nutrition program will play a major role. And in that, the life food, which is a very integral part for the any larval rearing program, which is must. Specifically, we have to go for the knowing the each and every species early requirement, like when they start to feed, what is the size of the live feed they can consume at the particular age, like from the day one or from the day five, and what is their nutrition requirement. Those need to be assessed, and in that different scientists are doing the work, like how to when their enzyme like pepsin, protease, lipase, they start to express, so they can give the exogenous feed to them. But before accepting the exogenous feed, we have to be depend on the live feed, because live feed is having the protein also, and they also have the enzymes, so those enzymes can help the digestion of that feed only. So in that way, we need it. So I hope I answered few of your points.
Yes, sir. Thank you so much. I also have another question, sir. Your work is
Okay.
also on reproductive endocrinology, no, sir?
Yeah.
So, how does understanding a fish's hormone and reproductive system help us choose which the right genetic traits for future breeding?
Okay. Can I ask you that what do you mean by right genetic trait? Because these are the two different aspects what I am understanding, to know about the reproductive hormones and the gene expression, and to go for this type of the traits. So can you much more elaborate your question?
I mean, we uh, we choose particular genetic traits to for future breeding. So, how
Okay.
does a fish's hormonal system or its reproductive system help us choose uh, this genetic trait, sir?
Okay. Nice question. So, like, if we want to go for the selective breeding, and we mostly in the fisheries science today, the concern of us is to talk about the, especially in the selective breeding, we focus on the growth related traits, and now up to some extent to the processing yield and also the disease resistant traits. So, for those type of the trait, growth related and the processing yield, if you can see that the instead of having the much more knowledge of the hormones and all these, if you have a marker related things, as like discussed by Dr. Suresh, that if you are having the link the uh, normal genetic or traditional genetic selection program with the markers, so it will help to accelerate the fast, to get the gain much more faster than the your normal selective breeding program. So that aspect will be the much more helpful. And if you can see that the reproductive endocrinology, the knowledge about the hormones and the genes and their expression, so which is the just a, primary things which you need to know in the basics also, that okay, these are the excesses for the reproduction and breeding in fishes. And if you want to do the work related to the reproductive endocrinology, so that can be on the different aspect also, which can go on the other sides, to know the explore the new hormones or new genes. Because what we know till now in the reproduction, if you go just 10 years or 20 years back, we much focus on the GnRH. Now if you can see, there are the lot of neuropeptides, like we already talk in the presentation, which are also having the several roles. So fish reproduction is much more complex, than we are thinking previously. So we can explore in that way also. For example, like in the fishes, synchronization during the breeding period is a very much issue. Like sometimes male are mature but females are not, or vice versa. So there is a issue in the synchronization of the breeding of the adults. How it is related to the hormones, how it is related to the genes, can be manipulated by changing the expression of the different genes or by giving some hormones. So that type of work we can think. So I hope I answer your question.
Yes, sir, you answered my question. But how can we use this in commercial farms?
Commercial farms, yeah, like if you have the knowledge of the reproductive endocrinology, like if you can see today the monosex uh, tilapia, so it is like a much more example from the commercial way. So if you want to do the monosex production of the tilapia, there are the different way, and mostly today people are using the that steroid hormone 70 to 20 beta testosterone. But the problem is that if you keep those fishes, or accidentally if the fishes are there in your pond for more than a year, they are convert back to their original sex and they start to again breed. So if you know the knowledge of this one, so you can manipulate on the other aspect also, like the genetical aspect, so that you can convert their chromosome itself. It's not the changing after that. So in that way, you can make a all male or all female, whatever you want. So you can do in a commercial way also.
Wow, sir, I actually did not know that. Thank you.
Welcome.
We have our next question for Dr. Suresh, sir. Um, while DNA barcoding is widely used for species identification, can it also help select genetically superior broad stock for breeding programs?
Yes, Aditi. If you ask me that question, yes, actually not. The purpose of DNA barcoding specifically it is for species identification. Means whether that species is correct or not. I would say no. If you want to select the fish for this one selectively superior trait fish, you have to use molecular marker, that robust molecular marker SNP microsatellite only, because it will detect this one DNA barcoding. We use only 655 base pair or CO1 region, that will give only that region whether changes are not happening. That can be used only for species identification, and diverse species you can select, but not for this purpose. We cannot use it. You have to go for microsatellite or SNP markers. Then previously I think one question, the conceptual difference, again, one point I want to add it, that the difference between eDNA and metagenomics. So when we use the eDNA, we collect samples from environmental, that is environmental DNA, either water or soil. We use primer for PCR only selected either for CO1 or specifically for 12S or something. So it will amplify only 700,000 base pair only. The name itself, it's a metagenomics. It will, we have to go for sequencing entire genome for whatever if you take one sample, you have to sequence for the entire genome for particular organs. With maybe many GB, you will get the data. But in case of eDNA, you will get the sample, but you will just amplify only the ports. For example, it is like if you go to the library, reading few books, that is eDNA. If you read entire library, that is genomics, metagenomics. That is how we should differentiate, conceptualize. I hope I have answered that. I think someone asked the last questions.
Yes, sir. Yes, sir. Thank you,
You
sir.
understood now?
Yes, sir. A part of metagenome is
Yes,
eDNA,
yes, yes.
as you
Aditi,
are
yes,
saying.
yes.
Yeah, thank you, sir.
Sir, you also talked about population genetics. No, how can we use modern gene mapping to, because nowadays everyone is using either tilapia or carp fishes. So how can we use indigenous Indian species for commercial production? Because there are many local species as well, more Indian species.
Means what do you want to ask finally, what is your questions? Means indigenous species should be developed into commercial. That is your point, or what is the exact question?
Yes, sir. How can we do that, sir? How can we do it sustainably?
See, sustainably, so anything comes, aquaculture comes means finally it's a market business, everything. So any farmers, the first trait comes as Dr. Pravesh said, it's a growth. Growth is most important, then followed by the disease resistance. Disease resistance is a very, very important trait for shrimp farming. If WSSP comes within 3 days, all the population die. So first, actually, growth. Faster the growth, better the performance, the more profit. Therefore, before, if we want to bring the indigenous species commercially, first of all, whether it is commercial or not, we should understand. It may be available in the indigenous, how many pieces are available. Whether that is commercial means what? It should be purchased by public, then it should be available everywhere. Then only if there is a profit in that, there is a marketability, then only we can go for the people will come forward to develop for the commercial level. Then what is the first step? That is aquaculture only. That is the captive breeding. You have to domesticate the fish at the captive condition. You have to establish the captive breeding. Then only you can go for another selective breeding program. Before think of genetic improvement program, you should have the captive breeding that aquaculture person must do, establish the breeding protocol for that particular fish. For selecting that fish, whether it is commercial or not, it is the local. Maybe one fish species may be commercially important. For example, northeastern, some of the fishes may be Tripura and all. Some species may be popular, may not be popular in Tamil Nadu level. So that should be developed in the particular regional level only. So some of the species only can be developed throughout the India or throughout the world. Even salmon is popular in other places. We don't eat, but in India also, the species should be developed at the commercial level based on the local interest, local commercial level. If it is, there is a market value. For example, in Tamil Nadu, something Tamil it is called Iramin, Leptocephalus thermalis, that species. It's a loach. That in Madurai district, sometimes it is sold 1 kg 1,500 rupees, 1,200 rupees. So what Tamil Nadu government said that now it has established exclusively one center for loach breeding program in Theni district. Now already they have standardized the breeding program and it will be disseminated to the farmers. First, it is developed at the commercial level, captive breeding condition in our university level, the center level. That will be transferred or transmitted to the interested farmers. Then it will become commercial. This is how it should go. That is what I think. I hope you have answered. If you have further questions, we can discuss it.
Thank you, sir.
Yes.
Before we discuss, I think Sagaya sir has another question for you. Yes, sir. Sagaya.
Sir, I have a question for both of you together. Pangasius is dominating in India. I think it is a fast-growing fish, single bone fish. That's fine.
Yes.
We have a Valago, or freshwater shark, which grows faster. When it comes to the cannibalism, even Vietnam they success with the marl selective breeding, marl, and they success with Pangasius. Why we didn't try for our Valago, which is single bone and air breathing animal? When you go for a selective breeding, because they prefer, if you compare both Maral and Pangasius, both are air breathing animal. Why don't we try for the replacement of Vietnam Maral? Why don't we go for a Chana Marulis, which grow 4 kg per annum? The growth is Chana Marulis is grow higher, double the time than the Desi Chana striatus. And other side, Pangasius replacement. Why don't we go for our Valago freshwater shark?
But if you ask me this question, why, I don't have answer. If you look at the China and Bangladesh also, they commercially produce in the freshwater itself more than 100 species commercially if you take. But in India, the last 50 years, we can still rely upon IFC, only Indian major crops. But why, if you asked, means it is who has to start? It is the investor or university or ICR Institute. It is there are multi-pronged approach is needed, sir. If anything commercial, for example, in Tamil Nadu, who all go for with the coastal plain only for shrimp. They will not switch into even the marine fish. They want to have the culture period short, and within 3 months something should be come out. Then within 3 months they should get some money. That is their motto. And if you look at this Vallagadu, I think still we have not developed the captive breeding condition and the seed rearing condition and commercial level scale. And but that's what your question. I understand why we have not developed. But some of the research captive breeding, like for you must have heard wompak fishes, sir. Wompak by Maclet, butter catfish. Yes, yes, that is popda. Yes, that is a very tasty, very nice fish. That is popular in north side also. Some Mettur Dam also we have that fish. Now last year, and we have Sottaival with Tamil, just they call it.
I know this.
Yes, that the Mettur Dam they collected and they have started captive breeding, but still the breeding happened, but the rearing stage, that is where that the freight to fingerling stage, feeding problem, cannibalism happening, that has not been done. So slow, it is being done, sir, but it is slow. But if you ask me the one word answer, I don't have answer for this question, sir. But it is the interest of ICR, and many people all must see exclusive institutes are there. You know, you are very well aware of that. SIFA for freshwater aquaculture. Exclusive work is for freshwater aquaculture breeding and aquaculture only. For brackish water, it's a Chennai, and so on for Nimtal for this one cold water fishes, and mariculture, semaphore. They are working, but what they have taken already species, that itself they are standardizing. Like for example, the Mukali fish, like recently they have developed. So these fishes, they have been developing fish, but it should be, I think, one by one. It is, yeah, we should take many more species. It needs a lot of effort, including manpower, facilities, all these things. And let's see, sir. Hope for the best. It will come out soon.
Thank you, sir. Pravesh, sir, you were saying?
Yes.
Yeah, I just want to add few points. And this one,
Yes, sir,
as
please.
Suresh sir told that we have a lot of species to work. India is very diverse in fishes, and I also believe that we have to work a lot because in our basket today, if we see that hardly farmers have the options for 10 or 12 species which are commercially available.
Exactly, sir.
We are doing the breeding. CFR tried and lot of success in the different marine fish species. Siva did, Sisifa did. But the exact thing is that it should reach to the commercial level, and it will reach when the farmers will stabilize their hatchery, like for the IMC, है like for the prawn, like monodon, and now for banana, like for the macrovacuum. Then only it can go. So yeah, our institute are doing the breeding in the different species they are touching, and if we come to the Volagato, that like it may come in their list, maybe in the last or just far from that, because it have different type of the feeding behavior, lot of carnivorous in nature, and so many things are there, might be. So that's all I want to add. And nice to see you, Suresh sir, after so much of the time.
What hurts most, sir? The problem is that we are growing everybody's baby, because I'm in the aquaculture sector, I'm into freshwater. There is a root channel, Paco, which is dominating now for the farmer's perspective, and the Benami, also the American product, the Nile tilapia from Egypt, it is dominating now. So the invasion, we call it an invasive species, right? Which is dominating. What happened to our own production, our own species? Because we are growing somebody's baby now. After 1950s or something, India is leading in aquaculture, because induced breeding is discovered in India, induced breeding. And after that, we haven't produced anything which is for global standard. I hope you both understand.
Yes, sir. Yes, sir. It is at the policy level. If you look at the shrimp, now it is Pinus Vanami, you cannot say no more it is invasive species, sir. It's Indian government analyzed with the SIBA and WFGN. They have done detailed research, and then they approved this species can be cultured in India. When we were struggling with the effect of white spot disease syndrome in the Pinus monodon, all were looking for alternative species. We did not have any species. Venus indicus was there. There was no growth, slow growth. Therefore, farmers need deliberately another species for farming in the coastal aquaculture. So what they thought, Venus, so all disease-resistant variety and specific pathogen-free variety were available outside. So America produced, many countries were keep it ready. Then what the government thought, government should help farmers. Then they were looking for some other alternative species. They formed the committee, NBFGR and SIBA, and all together work, CAA. Then they approved species. Now the export value of shrimp almost around more than 50-60,000 crore. Now, how can you stop this species? If you think also you cannot stop, you cannot call it invasive species. But despite that, now again some farmers have started culturing the pinnace. Yes. Now compared to means last 10 years, now it is picking up. Their demand is like prime species. We can say the rate is more, but compared to the just growth or quantity that production we take per hectare is compared to still less pinnace monodon. But it is so many lines are available. You name it. So I don't know. Yes, so many lines are there. They just at least once in a week in a specific pathogen-free line. This line is available. Fast line, slow line, that line, this line. So like that, there is we cannot say for our own monodon. That who has to develop, whether we or Asian countries or who has to develop. So these are the advantages. Those species that have advantage are thriving well. The farmers are also happy with the production and income generation. Within 90 days, it reaches its a particular gram, 20, 25 gram, 30 or 50, 60 counts. Then they get profit. So farmers' point of view, it's a profit. So whether which species, they don't worry about that. It's the government will decide which species, that's a policy level it should be made. So yes, it is that we cannot say significant level of pinnace monodon. Then it's it is picking up. Hopefully, even SIBA nearby only already is there. The NFDB has diverted around 20 crores for establishing selective breeding program in Pinnace indicus. It's our native species. So since you are asking today, this question was asked a long back, 10, 15 years back itself. Many people asked, why don't we have another alternative species? Only one species we have. So because they asked those questions, they answered. Now almost their hatchery work and selective breeding program facility is almost nearing completion. They have collected brooders also. They are one step ahead to produce selectively bred Pinnace indicus like Pinnace vanami. But still, if you compare current level, if you compare Pinnace vanami and Pinnace indicus, Pinnace vanami has more advantages than indicus and monodon. And still, farmers will go for this vanami only. But so this selective breeding program, hopefully they will make many generation and the growth rate will be superior than Pinnace. How many years it will take, I don't know exactly. So once it attains, definitely people will switch over that. Then next comes to tilapia. If you ask gift tilapia, we cannot say now it's an invasive species. It is like globalized species. There are many species. So specifically, it's a genetically improved tilapia. Gift tilapia only it is allowed for farming, but it is all invaded already with Vorechromis niloticus and everywhere all reservoir it is there. But tilapia is the most commonly cultured fish all around the world that cannot be avoided. But what one we can think, we have to go for alternative species, therefore no doubt. But if we go for alternative species, that should give profit to the farmers. That's the ultimate aim. So that should be developed by all, like university, ICAR institute, or including commercial farmers. That is how it should be. Hopefully, Pinnace monodon and Pinnace indicus in shellfish, it will come forward, and one day it will pave the way for Indian shrimp production, sir. That is my view, sir. Yes. Maybe Pravesh can add something.
Thank you, sir, for having me. That I have very limited experience of this shrimp farming and all this, because what I am doing here in Bihar from last 7-8 years, we are mostly working on the freshwater. But by knowing the things that I want to say that it's like obviously SIBA he start working on the indicus a lot from last 5, 6 or 10 years. They have did the selective breeding also, but it take the times, because the Vannami, when Vannami came in India It is not the overnight. It took the 5, 6, or 8 or 10 years to have the infrastructure, to have the farm adaptability, and to have the hatcheries and all these things. Now to switch from this Vannami to the Indicus or back to the monodon, obviously it again take the time, and also our institute need to have such type of the robustness in the species in terms of the SPF, SPR. They have to provide the option to the farmers, because farmers ultimately they are the businessmen and they need the profit. It's not like that we say, okay, this is the indigenous fish, you have to do this. No, 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 it's not like that. What I think, it's the that they want that, okay, if I take this species, what will be the growth rate, what will the FCR, how they are resistant or not. So all these things matter. So first of all, institute need to work on the breeding aspect, on the selective from breeding program, and then need to change the farmer perceptive. We need to make aware that, okay, our species, native species, can also perform uh, with same like the Vannami or at par from the Vannami also. So I hope this required. Thank you.
Thank you so much, sir. Because the reason that I put this question, both of you, that I was thinking about the sales aspect, because you both speak about the farmer aspect. When it comes to the sales aspect, Pangasius prices today it's in Andhra and Tamil Nadu it's 75 rupees per kilo. So when it comes to the feed cost, it is very difficult to sustain. First point. That is why we need a diversity, because customer want different species to eat. See, and the Vietnam model, we already have a problem that both, I think, you have studied my newsletter about something. I wrote about a newsletter. It's about something. Vietnam model. The Vietnam model, the problem with the Vietnam model, I have been into many farms in Tamil Nadu. The Desi and the Vietnam, the selective breeding or pure breed, got mixed in the wild, because Chana striatus to reach 800 gram to 900 gram, it takes 12 months. When it comes to the Vietnam, it reaches in 6 months. So what happened now, when it is introduced to the wild, Desi grows slow. The population, the diversity, the mola, the Fintius, other species are moved. But this Vietnam, it grow fast, it predate fast, it collapse the environment. I have seen literally in farms here in Tamil Nadu, in the lakes, everywhere. I'm getting in 1 year 2 kg, 2.5 kg of Vietnam in the lakes. It is completely disturbing the environment. This is another perspective when it comes to the genetic invasion. So the problem that the consumer aspect, it is very hard. We need a different variety. Roots and price here, it is today 90 rupees. Roots and price is 90 rupees. And tilapia you can get at around 50, 60 rupees. So how can we go for the feeding at such a low price? So we need some alternative. This is why I put this question, because I was doing this business more than 78 years now. I analyze the market. Now, predominantly, I work with Kerala, Tamil Nadu, Andhra, Karnataka. I supply fishes to domestic in the 4 states. We could not even sell a 45 rupees to tilapia to the wholesaler today. This is the present scenario in Tamil Nadu, in Kerala, and Andhra. So we need some option. We need some variety. Why we need to rely on this Katla Rogu and other stuff? Why don't we need some for export premium? When you go for export, we compare with Vietnam, they succeed with Pangasius. The only fish, they supply globally 80%. Vietnam. So why don't we make a premium one, our Valago? This is the way I just was thinking quite long. So what is the replacement for my farmers? And I was studying about Valago. It grows, Vietnam grows 2 kg in 1 year. Valago also grows 2 kg in 1 year. But in 2 years, in 24 months, Pangasius will grow only 3 to 3.5. But Valago goes to 6 kg. Valago can grow up to 6 kg. So it grow fast and single bone species and air breathable. We can easily compare. So and also it is highly nutritious and highly premium. So instead of selling at 80 rupees price, why don't you go for a premium one and we give option to our farmers and our consumers. This is why I narrated these things to with you. So this is why we need a variety, not to rely on this. Only in sales aspect also we need to think. Because now what happened to shrimp industry is completely collapsed. I hope you both understand my question.
Okay. So what I think that like here, if I just thinking about the Bihar also, when you are talking about the Tamil Nadu, that the prices of the pangasius go up to the 70, 80 rupees. But if we see here in the Bihar, the prices of the pangasius is 150 rupees. If I see the prices of the IMC in Bihar, it's like a for the life fishes
One
of roe, it's a 250 for the katla 300. So what I think that obviously whatever you highlight it's important that we should have the diversity, we should have the diversity in the farmers bucket, that whatever the fish they want to choose they can do. But for that obviously work is going on and different research institute are actively involved. But as you know, sir, that it's a long process to domesticate, to acclimatize, to breed, to larval rearing, all these aspects, and finally to the commercialize to the farmers to have the large commercial structures of the hatchery. But what I observe here that it's a issue of the marketing also. It's a issue of the processing aspect also. It's a issue of the demand and supply also, because like here if I can see the pangas, it's 150 on an average from the farm, and from your side that you said 70 to 80 rupees. So I think the processing infrastructure then required, because if the production is high, demand is low, the prices are crash. So for that we should have the other options for the preservation of the fish, for the other marketing channels, for the processing aspect, what I think. And the certainly you said that we have to focus on the other fish also. Obviously it required in India we have very diverse type of the environment, different places people have the different preference, different type of the fish they prefer, so we should have that. And we are doing also here, we are doing, and there are the different institute they are trying to do the local fish which are indigenous there, like I have shown in my presentation that we are doing for the magur, we are doing for the gechi, now we are uh, planning to work on the small indigenous also, which have some ornamental value or some maybe for the food fishes which are very small in size, so that in Bihar people have the practice to consume the whole fish also, like without piece and all these things, just a very small fish of 10, 20, 50 grams. So they just remove the scale and intestine and then fry it, and it can be used. So it is a good source for the minerals also, obviously. So that is we are doing here. We are working on the breeding aspect specifically, because it's a must in aquaculture. The diversification is required, whatever you said. That's all I want to say.
Thank you, sir. Thank you, sir, for your information. I've been doing business in northern part of India as well, but problem the pricing are higher. And in North India, the farming is increasing. Now the real question is that the South India we don't have market. North India they relied on our fish. Now when they started producing their own fish, now our market is got collapsed. Now farmers are in southern part of India. They are leaving aquaculture industry now. I think in past 2 years, almost 30% of people they left this industry. It's something like if your pangasius price is today 80 rupees, when it is reached to the Bihar, it will cost us around 110 rupees or 120 rupees. The ice fish, the 40 rupees or 30 rupees goes for the transportation for the 40 rupees. So 120 it will go, or 110 it will go. The another margin add up for 10 rupees for the wholesaler or 50 rupees for the wholesaler. It is equals to your domestic price. It comes to nearly 130, 140. So still the farmer money is only 80 rupees. Transportation kills minimum transportation ice kills minimum 30 rupees because of the distance.
So yeah, as like you said, sir, that if the south start to production and they have the market crash, so it's need
Yes, they have
they need the specifically the
alternative market.
processing and the export and all these type of the markets they have to explore, because I'm not much knowing the feeding and all these habits in the South India, but what I assuming that it is preferred for the coastline, so they have the preference for the marine fishes might be, and the marine production is also there. So the freshwater aspect, then the production is enhanced, then the market is collapsed, demand is low. Maybe that is the reason
Yes.
you can correlate much more easier. So apart from what I want to say, apart from to have the diversity in the species, they also need this type of the export market and all these things. Because today what we see here, that the Pangas, Pangasius, and then
Yes.
IMC. Even today, this Paku, they are coming in Bihar all along from the Andhra in the ice
Yes.
frozen form. So what we can see that the fish from the Andhra, as you mentioned, it have the transportation cost. Obviously, if it is travel from 1,500, 2,000 kilometer, it have the cost. So it's there, it's there. So it need to explore the other aspect, other markets, and other type of things also.
Yes, sir.
That's
The
all I
best
want to say.
thing.
Thank you.
Yes, sir. Thank you, sir.
Nice to interact you, sir. Thank you.
Thank you.
Thank you so much, sir. I think we're in the very end of our session. Before we end our session, I would like to ask only a final question, small one. Um, we all know that in genetics, genetic bottleneck is like a biggest issue. So how do you think, like in aquaculture, how do you think we can fix this genetic bottleneck, sir, to help diversify the genetic health of the fishes?
It's being a genetician, I am having the my work in the genetics and specifically breeding. Whatever I can see here, the hatcheries in the Bihar or the North India specifically, even in the Maharashtra, I have seen that the problem is that it's not profession. Means they are not the professional. Whatever the hatchery owners are there, they are not care about the fishes, because what they think, okay, if I breed one pair in IMC, if you can see, lakhs of these spawns are there, right? And
Yes,
if
sir.
they breed one pair of the kettles, you can see hardly one calf or two calf. So they are much more bothered for the their kettles to feed them, to take care them, to have the progeny, because it give hardly one or two calf, one or two children only, na child, young ones. But here in fishes, lot of eggs are there, lot of spawns they get. So they are not much bothered. They are not bothered for the brooders to have the proper feeding, to have the proper size of the brooder. No, and they are not much bothered to have the stock size, because if I say the fecundity is very high, so what they are doing, they are breeding the same fishes again and again, and that cause the inbreeding, then inbreeding depressions, and then the slow growth rate, and a hatchery which start to produce the IMC seed. In the initially, farmer will say, it's a general, farmer will say, feed is seed are nice, they are growing very well. But after 5 6 years, they start to complain. They are saying, no, उस hatchery का seed अच्छा नहीं है. There is no, it is not a good seed. It is not growing. In a year, it growing hardly 500 gram or 300 gram on an average. So the problem is that with that hatchery owner, with that the manager who is at the hatchery, that how they are managing their brooders, how they are take care of the brood stock, how they are changing the brooders, what type of brooders they are selecting. So it's a very professional. They need to be professional. They need to know about the genetic aspect. They need to know about the inbreeding. They need to know about the importance of having the large stock. And till now, what I see, very few professionals are there which are involved in the fish seed production, or especially for the IMC. I'm not talking about the shrimps and all these things. Obviously, it is there, but in the IMC breeding, what I observe, it is the issue, and mostly the owner is not very much concerned for that one. They think that okay, just hatchery बन गया and उसको किसी को दे देंगे और अच्छा खासा प्रोडक्शन होगा. Professionalism is required, what I all want to say.
I understand, sir. Uh, as a fellow genetist, I understand completely about breeding and everything, and how genetic bottleneck is like the biggest problem. I understand what you're trying to say.
Yeah,
Thank you so much, sir.
thank you, thank you.
Yeah. So now we are like at the very end of our session. So I would, as our session has come to an end, I would like to heartfully thank to all of our distinguished speakers, Dr. Pravesh Kumar.
Thank you.
Thank you, sir. Thank you for reminding us about food security, for synchronization, for fish breeding, and everything, sir. Your words mean a lot. And Dr. Suresh Eswaran, thank you for revealing how reading the genetic code can build a more sustainable future. From a molecular code inside a cell to a young fish's first day of life, today's speaker have shown us that a future of fisheries is being written at a scale most of us has never seen. To our speakers, thank you for your generosity, your expertise, and your time. And to our audience, thank you for your thoughtful questions and engagement. You are the reasons conversations like this matter. Over to you, Akansha.
Thank you so much, Ki. So now we see that this brings session 8, the DNA of tomorrow, to a close. Thank you once all. Thank you all once again for being the part of this conversation on the future of fisheries and aquaculture. Please stay with us for our next session. Seaweed is not just seaweed. Thank you so much, everyone.